because the agency the agency that handles this type of revision process is having so many other works also. Uh, what is required to revise the syllabus? Of course, when the syllabus is to be drafted or revised, the concerned board of studies in the subject appoints a subcommittee, and these subcommittee members in their various meetings finalize the syllabus. This subcommittee has to prepare an outline or the skeleton of the syllabus right from beginning, that is first year, up to the final year. They have to decide what should be the number of papers in the syllabus, what should be the titles of the papers, what number of lectures are to be assigned to each of the paper and each of the unit that is included in the syllabus. Unitization of the syllabus, that is very important. Once the unitization is done, then the syllabus committee has to go for detailing of the content of the syllabus. While carrying out detailing, of course, it is necessary to specify the points that are to be covered in the syllabus units. This is very important because the same syllabus is being implemented by a number of colleges or the institutions in and around the university. In order to have the uniform structure and uniform teaching, it is necessary to specify these points which are to be covered in the unit of the syllabus. Now, what's the process of the syllabus revision? The first step is drafting of syllabus. As already cleared, that the agency that is involved in this syllabus drafting is Board of Studies in the concerned subject, which appoints the subcommittee, and this subcommittee in number of meetings will finalize the syllabus. Then this syllabus that is prepared by a subcommittee has to undergo certain administrative processes. For example, such a syllabus, first of all, is to be presented in the Board of Studies that will finalize the syllabus, that which then will go to Faculty of Science. In its meeting will approve the syllabus, and the final approval is to be obtained from the Academic Council of the institution or the university. Once this all administrative procedure is complete, then the next work of the institution or the university is to train the faculty members by organizing the workshops, syllabus workshops. In these syllabus workshops, the expert lectures are to be organized that will throw light on this syllabus. They will discuss about the depth of the syllabus that is to be given to the students. Then the teachers' participation will be there, that what should be included, up to what depth it is to be gone through the syllabus. And finally, the doubts that are raised by the faculty members or the teachers in the workshop are to be clarified by the concerned experts. Once the syllabus workshop is over, then it's our duty of the teachers to go to the students for its implementation. As we know that for syllabus implementation, three things, they are very important. First is the teaching process. This is followed by learning process. Of course, this will be simultaneous one. And at the end of the teaching and learning process, have to go for evaluation of the students to judge their knowledge that we have given to the students. We teachers, we have a major role in teaching, because the interest is to be created amongst the students, along with giving the thorough knowledge of the subject to the students, so as to have their better and the bright career in the future. While teaching, the role of teacher is very important. As I already have said, the teachers should use teaching aids in order to deliver a better knowledge to the students. And these teaching aids will include the preparation and presentation along with models, along with charts, the PowerPoint presentations. One even can share the educational videos that are prepared, which actually are available on YouTube. Or once the teachers, they can prepare their own educational videos. They can construct their own YouTube through which the students will get the thorough knowledge. In addition to these different teaching aids, It is necessary to go for group discussions. And of course, teaching, it's not a one-way process. It is necessary to involve the students, and when we'll have the interacting sessions or interactive sessions with the students, of course, with participation, they will get a better knowledge. Learning process, as already cleared, is a simultaneous process along with teaching. And it's our duty to develop interest in the students. It is necessary to make easy learning process with lucid, fluent, and the informative teaching. It is necessary to clarify the doubts that are raised by the students in the lecture itself. It is also necessary to make the students knowledgeable rather than exam oriented. This is mainly because even though the examination score is very important in order to get a job, to keep the job, of course, the knowledge is very, very important. In the lecture itself, it is necessary to ask the students to refer the reference books that are available in the library. They also are advised to prepare their own notes or own articles. We have to make them more practical oriented rather than the theoretical knowledge. Once the teaching learning is over, we have to go for the process of evaluation. Evaluation of the students can be done by a variety of the ways, that one can organize different seminars, one can organize group discussions. We have to go for the continuous internal evaluation where we have to conduct different tests, tutorials by asking the multiple choice questions or essay type questions with a time limit. Once the tests or the tutorials they are over, then it is necessary to evaluate these all papers, and after evaluation, each and every student should be allowed to present yourself and ask and discuss in detail about the performance. So all this is needed in the syllabus revision of the subjects, not only for microbiology, but it is also applicable for all the subjects. With this brief, now let us go to the syllabus that has been revised for PSC Part 1 Microbiology, that is to be implemented from August 2021. Of course, syllabus, it's very good, neatly organized. We have taken the capacity of the students into consideration, and the committee has prepared this syllabus in a very lucid way. As we are aware that each of the course or the paper will have 4 units. The course 1 or the paper 1 of BSc 1 semester 1 is introduction and scope of microbiology, where the unit 1 includes the discussion regarding history and development of microbiology. One point that is mentioned in this particular unit is contributions of the scientists in the development of microbiology. Because of the time constraint, of course, the contributions of the specified scientists are asked to elaborate. Of course, these scientists, all the scientists, they have not been covered in this, but because of the time constraint, as I already have said, that it is not include all the scientists that are involved in development of microbiology. The unit 1 also discusses, or it is necessary to discuss, the theories of biogenesis and abiogenesis. So unit 1, it's okay. For unit 2, where have to discuss different branches of microbiology, they have been specified in the syllabus itself. What I will suggest that in the syllabus it is given as air microbiology, water microbiology, sewage microbiology. We have to give a title for this as the environmental microbiology, and under that you can give the subpoints as the air, sewage or water microbiology. Then there is environment of agricultural microbiology, food and dairy microbiology, industrial microbiology, medical microbiology. What I suggest in this particular unit is that it is necessary to make mention of geomicrobiology, where I have to discuss in short what's the role of microorganisms in the leaching of the ores or extraction of the ores from raw material. It also should include virology. To some extent, have to give some explanation for this particular branch which is very prominent and is very important in the current scenario. Have also introduced about space microbiology or even in the petroleum microbiology, because microbes, they play a very important role in formation of petroleum and the petroleum products. Not only formation of petroleum is of microbial origin, but recovery from oil wells in seashores also is having an importance of microorganisms which will be helpful in secondary recovery or extraction of the oils. Unit third, it discusses introduction to types of microbes, which will include differences between prokaryotes and eukaryotes, different general characters of microbial groups, and bacterial nomenclature and classification. This unit is okay. Unit 4 is regarding the scope of microbiology. Actually, scope has already been given in wide details, but again, it is necessary to have some additions in this unit also, because microbes that are not important in the environment, agriculture, food, dairy, medical, and so on, but they are also very important as far as the research field is concerned. So basically, the research is done on microbes. So we should elaborate to the students that how and in what way these microbes, they are much helpful for the research. Then how these microbes, they are helpful for exploring or searching oil fields on Earth's surface. How they are important in leaching of ores, and in what way they play a very important role in the space study. So that was about the first paper. Then there is second course, that is of bacteriology, which again has 4 units. Unit 1 is bacterial cell organization, where the students are to be given knowledge regarding the morphology, structure, chemical composition, and functions of certain anatomical parts of the bacterial cells. Unit 2 is microscopy. Actually, in this microscopy, number of microscopes, they are to be introduced. At least students should know that what type of microscopes they are available for the study of microbiology. And of course, the much stress is given on light or the compound microscope and the electron microscope. This particular unit includes all the basic knowledge, for example, the numerical aperture concept or resolving power of the microscope and so on. So all this basic knowledge regarding microscopy is to be given, because it becomes a very important task in the techniques for study of microorganisms. Unit 3 also is very well explained, that is stains and staining procedures, where it is necessary to cover types of stains and even the different types of staining procedures. Unit 4, that is very important, that is control of microorganisms, because this is the key process in formation of different products on commercial scale. So it is very important as far as the industrial microbiology is concerned, and many of our students, they are going to the industries for their jobs. Some are involved in the production units, some are involved in research and development wings. So it is very essential for all these students who are going in the industrial sector that they should have the knowledge of control of microorganisms, because in the research field and even in different industries, the aseptic conditions and the pure culture work, these are two very important processes. That topic also has been properly given the justice, including the physical things or factors and the chemical factors for control of microbes. When we'll come to the practical of semester 1, the practical, it's okay. Especially in the practicals, it is necessary that one practical is there, that is in the list of the practicals, that is preparation and sterilization of various media, where I want to suggest one thing, that in this there is make mention of only certain media, but it is necessary to introduce the preparation and sterilization of peptone water or starch agar, milk agar, sugar media. So this is to be included. And there is one more thing, that the anaerobic jar that comes under study of common equipments, it is a repeat in practical 1 as well as practical 2. So that repetition is to be avoided. When we speak of the practical examination, at the end of the syllabus, the structure is given regarding the practical examination, where in the question 1, it is a determination of efficacy of alcohol, copper, or sanitizer. Actually, in what way these practicals are conducted, that is to be seen. And in addition to this testing of efficacy, one can even go for testing efficacy of sterilization by autoclave or sterilization by use of ultraviolet radiations. Actually, it's a process of disinfection. So one can check even the efficiency of UV disinfection process by adding it into the practical. In the semester second, course 3 is regarding microbial physiology, where again 4 units are there. Unit 1, microbial nutrition, where we discuss nutritional requirements of the organisms and the nutritional classification on the basis of their nutrition. This unit is perfect. Unit 2 is culture media, common components and types of media. It also is okay. Unit 3 is bacteriological techniques, isolation of the bacteria by using different techniques as pure culture techniques, that includes the strict plate, spread plate, and pour plate. Maintenance and the preservation of microbial cultures, that also is very good one. And unit 4 is systematic study of pure cultures, where the bacterial cultures or the pure cultures are to be studied with respect to different characters as morphological characters, cultural, and the biochemical characters. I want to make one suggestion in the testing or studying of biochemical characters, that in addition, one can add in biochemicals the testing of ability of organism to produce metabolites like indole, or quantity of acid, or ability to produce acetone by the bacterial cultures. So one can introduce IMVIC in short, that will be helpful even in the examination, while conducting the examination. Then in the second semester of PSC 1, there is a fourth paper or the fourth course, that is biochemistry. This topic or this paper is very nicely and neatly designed, so it is not necessary to go for any comments on this. Regarding the practicals, as already cleared, that in the demonstration of laboratory equipments, it is necessary to avoid the repetition of anaerobic jar. I have already mentioned that it is necessary to include preparation and sterilization of certain media as peptone water, starch agar, milk agar, glucose phosphate broth. Because starch agar, milk agar, we are going to use it for testing ability of organisms to produce enzymes like amylase, casinase, and so on. The checking ability of bacteria to produce indole, acid, acetone, that also can be included in the syllabus if the authorities permit. As far as the practical examination of semester second is concerned, in the format that is given for conducting practical examination for semester second, some discrepancy there in the marks. Question 1, it should have 10 marks instead of 15. So that is to be corrected. After covering this BSc 1, semester 1 and 2 syllabus, now let us go to MSc part 1, semester 1 syllabus. I have gone through the syllabus, theory papers 101, 2, 3, 4, and practicals 5 and 6. All this syllabus that has been mentioned in MSc 1, it's okay. One suggestion will be there regarding the course 2 of the practicals in 106, that already we have mentioned about the practicals of estimation of amino acids, proteins, nucleic acids, that is DNA, RNA also. But if possible, one can go for including the estimation of lipids and the carbohydrates, because these two points, they are covered in the theory. So at least one method one can ask for the students to carry out estimation for lipids and the carbohydrates. Regarding the syllabus, as already I have said, that the syllabus, it has been designed in a very specific way. It is neatly organized. It will have the continuity. And of course, when we are going to revise the syllabi of second year or the final year of MSc, we have to consider these syllabi in order to avoid repetition. Or if there is repetition in the previous years, if it's just a mention, one can go for the detailed study of these topics in the following or the subsequent years. I think this is my opinion regarding this syllabus. If the microbiology faculty members who are participating in this, if they will have certain queries, then they may ask. I will try my level best to give the answers to such queries. Hello.
Yes, sir. Now session is open for discussion. Participants, you can ask questions regarding the syllabus. Okay, if there is no questions, we can move on. Thank you very much, sir, for giving valuable insights regarding the BSc 1 and MSc 1 microbiology syllabus. Now, I would like to request Mr. B.S. Pawar, sir, to propose vote of thanks. Over to you, sir.
Thanks, ma'am. Hello, everyone. I, on behalf of Microbiology Department, I would like to thanks today's resource person, Supnikar sir, to take out time from his busy schedule to grace the event. I would like to thanks our beloved Principal Dr. B.T. Jadhav sir, our Vice Principal Deshpande sir, and all staff members. Once again, I thanks one and all present in online platform. Thank you. Over to you, ma'am.
Thank you, sir.
Oh, thank you.
Thank you, Supnikar, sir.
Thank you. Thank you very much.
Now, I hand over the charge to Dr. Ganesh Jadhav, sir.
ओके, आपला आता नेक्स्ट सेशन चालू होईल आता फूड प्रोसेसिंग डिपार्टमेंटचा. हॅलो. So the next session start by the food processing and packaging department.
Good afternoon, everyone. I welcome you all to the second session. So I am here to introduce our resource person. We have one of our faculty member, Mr. P.R. Dure sir, who is going to talk upon the recent trends in food processing and packaging. So before he begins, I'll just brief about him. So he has completed his BSc in Food Science and Quality Control from Shivaji University, Kolhapur. Then he has completed his MSc in Microbiology from Swami Ramananda Tirth Marathwada University, Nanded. He has also qualified NET exam in 2019 and is also pursuing PhD from KBC North Maharashtra University. Talking about his work experience, he has worked as a production manager Blue Star Agro Winery Private Limited, Chakan, Pune. Then he has also worked at Satya Vijay Institute of Research and Management. He has worked as a principal of College of Wine Technology, Saswat, Pune. Currently, he is working as assistant professor at Department of Food Processing and Packaging. So I request Mr. Dure sir to begin his talk. Over to you, sir.
Thank you, ma'am. In the beginning, I will thank to Dr. B.T. Zadav, sir, the principal of Yeshwantrao Chavan Institute of Science, for giving me such opportunities. Also, I express my thanks to Dr. S.S. Wadikar, ma'am, giving me this opportunity to deliver my views on recent trends in food processing and packaging. So before proceeding recent trends, we should know the scope of food processing in India. So all these departments are related with life science, so they are very well known the scope and diversity of India. So India have regional and cultural diversity. India has about 26 types of different climatic conditions and near about 46 varieties of soil. All over the world, there are 60 types of soils are present, and out of that, 46 types are present in India. And again, India is divided into 127 agroclimatic zone. And so, as this is a drastic change in its climate, soil condition, so it definitely gives us the benefit for producing a variety of fruits and vegetables. So, the Indian food is also known worldwide for its unique taste and aroma. So, India produces nearly about 16% of world's total food grain production. And so everyone knows that the day by day, the population get increases and the demand for food is again increases. And it is estimated that 590 million people will be the population of India by 2030. And so that it require that much amount of food, and so that much amount of food we can't produce in a whole year. So we should preserve that food. And so this is the scope of food processing. So when we when we go through the economics of that sector, so everyone knows that the growth rate of India is before COVID situation is 6.5%. The growth rate of India is near about 6.5% before COVID situation in 2019. Then after in COVID situation, it dropped down. And at the same time, the growth rate of agriculture sector is 4%. But on the same time, the food processing industry grows with 8% per annum. So this is much more higher than agriculture and other field sectors. So the COVID-19 affects every field. We know that it impact on every field, and definitely the food processing sector, agriculture sector, every sector suffer from COVID-19. But so this is the common thing we talk about every sector, but the sector which is remain aside from this impact, that is the food processing sector. Before COVID situation or before COVID status, just I again put some figures. The growth rate of India is 6.5. The agriculture growth rate is 4%, and food processing growth rate is 8%. During COVID situation, the growth rate of India declines from 6.5% to 0.4%, and then after -9%, -0.9% during the COVID situation. And then after post-COVID, when we think on that, so it is now start to grow up near about is in 2021, the growth rate, March 2021, growth rate of India is 1.3% now. Still, the growth rate of food processing industry remain as it is. Pre-COVID situation 8%, during COVID 8%, and post-COVID again it is increases to 8 to 9.5%. So this is the scope of food processing. And when we think on the food processing sector, it contains different sectors like fruits and vegetable processing, seeds and grains, marine products are there, meat products, and milk products. So this is the segmentation of food processing in our growth rate or in the country's growth rate. Next, so this figure itself indicate the scope of food processing. Now, due to COVID, everyone is thinking about thinking on the nutraceuticals. There is an increasing concern for the impact of food habits on health and growing needs for essential nutrients for the healthy lifestyle. And with the COVID-19 pandemic situation, now consumers are focusing on the eating healthy, again making nutraceuticals. So this is the field. This is now a recent, we already studied nutraceutical. Now we have to study nutraceutical along with in the light of COVID-19 right now. So it's again through that scientific research, through that the nutraceutical of various types, it provides the health benefits for against the disorder related with the oxidative stress, then allergy, diabetes, and immunodiseases. So there are some more starters are started in the nutraceutical. This is the recent trend in the after COVID situation. Nutraceutical, we already studied nutraceutical, but now we have to study, we have to focus nutraceutical in the light of COVID-19. So these are some outstanding or out-of-box startups in the nutraceuticals. That is the Farmhand Organics, which produces the probiotics, prebiotics, and the postbiotics to improve the immunity balance of the human being. Then these are some sectors of food processing in which the students will get the job opportunities. Just I go through that slide. The fruit processing, everyone know that fruits and vegetable, it's a very large sector than food grain. That we will go through one by one in detail. What is the scope of food processing in a particular sector? The processing of milk and milk products. When we think on milk and milk products at that time, India is the world's largest producer of milk, and out of that, just 2% processing is carried out. Otherwise, all the milk which is produced, it is sell out as a raw, and there is no that much processing. But we have to increase the processing, and then after the food safety by this processing. So this is just 2% processing is there. So anyone can imagine there is a lot of scope for the students in this sector also. Then we are thinking on the inflammation day by day, and we are also thinking on the rate of increasing the prices of petrol and diesel. On the same time, there is a large increase in the prices of edible oil also. Why such type of the condition is arise? So there are some agriculture factors also and some processing factors also involved in that. So there is a single cropping pattern is arising all over the India. Every farmer is thinking on the cultivation of sugarcane or the cash crop. And so that the day by day, the land we under the cultivation of oil seed get decreased, and which impact is now we observe in this sector. So the rate of inflammation related with the oil, edible oil get increases in the before few months. The soybean oil for 1 kg is near about 130, and now it's 110, and now it's comes to the 135 rupees per liter. So this is the processing and along with the production. When we think on the per capita consumption of oil seed is again increased 3 kg in 1950, it's increased to 6.4 kg in 1996 to 1997, and in 2016 the per capita per year consumption of oil seed again increased due to its the changed lifestyle of the human being, especially from India. The consumption is increased from the 6.4 kg per year per person per capita to 18.5 kg. And on the same time, we should again note the recommendation, WHO recommendation for the intake of oil seed, and this is the intake of this is the recommendation of WHO for the consumption of oil seed, that is 11 kg per person per year is required. And we again, so that the demand is increased due to our consumption is also increases, our population is day by day increases, and the area under cultivation is decreases. So today India is still importing around half of its domestic consumption. The domestic consumption, the required quantity and the needed quantity, it's there is a, we have to import 0.5% of our domestic consumption. Next, we will move towards the next sector, that is processing of cereals and pulses. So this is the data from 2018-19. Total food grain production, that is 284.95 million tons. So this is in that sector also processing is carried out, proper storage is required, and for that proper storage we are always listening the news like India have the grains or the cereals and pulses stock for so as to maintain our indoor demand for the next 2 years. And so five such type of the statements are come. So the cereals and pulses which are produced in India, these are well stored through the FCI department, that is the Food Corporation of India department, under the central government. And then the processing related with the cereals and pulses is as well, everyone know that the instant food, that is a bakery. So there is one term normally we think on that is a bakery and confectionery that are not produced as fast as they consumed. And so as a result of that the day by day the demand from the bakery products is increased, and so the students will get the opportunity as an entrepreneur in this sector in the form of the wheat flour, then sugar, sugar, water, ghee. Such type of the raw materials is used for the bakery, and after applying some processing steps like baking, mixing and fermenting, molding, baking, all this we will get the bread, buns, biscuits. So everyone know that. The next sector is the processing of meat, fish and poultry. Again, India is the largest population of the livestock in India, and near about 5.3 million tons, metric tons of meat is produced annually. On the same time, 75 billion eggs are produced annually. Out of that, when we think on the processing of such type of the meat and fish, when current processing level in poultry are the 6%, while for the meat it stand for 21%, and the processing level for the marine products in India is near about 23%. So there is again scope for the processing. And now the field, the recent field or recent trend in the meat, fish, and poultry processing is that the animal welfare. So animal welfare is very important. Its health status, level of stress is again very important, so that whenever we slaughter such type of the animal and the nutrients from that we get, so there is a proper hygienic conditions required, and all this consideration all these points taken into consideration by some industry like the Saguna chicken, then Venki's and Baramati Agro, they are focusing on exporting the meat and poultry. So this is the export potential for the meat. Near about 90% of meat obtained from the buffalo beef, which is exported to the other countries, and again 3% the meat from sheep and goat is exported. So there is a large scope for the exporting such type of the meat, fish, and poultry after processing. So APEDA is the Apex Institute, which control all these export activities. There's agriculture and processed food export development. And then there are 33 export-oriented slaughterhouses are there, and 35 meat processing and packaging units are. This is the data collected in 2019 all over the world. So we can imagine the scope of export in meat sector. Now we'll move towards the spices processing. So India is known as the home of spices, and so that in 18th century, the British India Company come for the trading of the spices, and they again convert as emperor of the India. So everyone know that this is only due to spices. So this is the spices, and near about 45% the total production of spices globally is from the Indian origin. And again, ISO, according to ISO, that is International Organization for Standardization, there are 109 spices are present all over the world. Out of that, 75 spices are produced in India. So this is only due to a wide variety of agroclimatic zone, diversity in the soil, and also there is a climatic conditions, and all these get benefits. All this gives benefits for the food processing sector. So this is all about the processing of spices, and now we move towards the new technology which are coming to the food processing sector. That is a high pressure processing. So for avoiding the non-thermal processing, we have to switch to the high pressure processing for the preserving the different food products. And due to application of the thermal processing, there is a loss of nutrient also, and there is again loss of some amino acids which are present in the food. So this is a new technology, which is a recent technology in the food processing, that is high pressure processing. By using the high pressure processing, we can preserve different food products like ready-to-eat curries and also meals. Some starters are again started in this sector also. Now, as I already told you, due to COVID, the consumers are again shifting from one source to another source. That is, alternative proteins are again required. We know that the mycoproteins are there, but in the same way, the cultured meat plants are now. This is the trends get started. Cultured meat plants, edible insects are again there, and some starches are again started in this sector. That is alternative food proteins. So this is the recent technologies which are comes into the food processing. Now, food safety and food transparency. Everyone know that food safety, but along with that, the food transparency is very important. In that, end-to-end traceability. Whenever the food is packed, and after that, some changes is carried out as time passes. So there are some sensors are present in the food packets, and it indicate the level of deterioration of that food product. It also indicate the changes in that food, especially in case of nutrient loss. All these changes easily denoted by this technique, that is a transparency, food safety and transparency. In that, end-to-end traceability is there. So there are again some startups get started in this sector. That is a Quality Automats from the French, and which are working on this new technology. Now, day by day demand for the fruits and vegetable grains are increases. So farmers are adopting the different practices for increasing their whole production. In that they are using a pesticide, weedicide, and so that residue remain in the food. So now, and so such type of the residue remain in the food, it may be carcinogenic. So that now consumer are that much aware, they should, they know, they are aware for the such type of the carcinogenic substances. So we should quantify the risk and protecting the consumer from the pesticide residue. So this is one another sector in which recent trend is carried into quantifying the risk of the pesticide which are present in the food products or the processed foods. So this is the some general formulas are used for that, and by that formula we can easily quantify the risk of the pesticide in our processed food also. Now this is all about the recent trends in the food processing. So there is one another sector, that is the food packaging, and our the course title is food processing and packaging. So this is always a question for the question of debate. There is a unique, the title of the course is not that much unique. It may be food science and technology, it may be food technology, it may be food processing technology. So title, there is a sometime our home university peoples are also thinking about that, but I will focus on the what is the scope of that packaging. Indian Institute of Packaging present at Mumbai, it offers 2-year postgraduate diploma in packaging, and then after the YCIS Satara offers such type of the packaging. So YCIS, our institute is the 5th institute we offer such type of the course in the packaging, especially in food packaging. So from this year we are running the MSc for in the food processing and packaging. So we concentrating the packaging. So such type of the papers are added in our syllabus. 103, introduction to papers, paper, paperboard, plastic and polymer. Then 203, the packaging law and regulation. 303, advanced food packaging. Then 403, packaging design, concepts, application of that packaging into the application of computer in the packaging design. So this is the scope of packaging. So many times people are talking about, or some students and some people are passing comments about our title, that is a food processing and packaging. So this is a unique, this is a new trends, packaging. So we are the 5th in our country which offers such type of the packaging course for the UG students, and along with the UG, now we are offer same packaging for the PG student also. So this is the new trends for the packaging, labeling, marking, then other food product identification machines are there, then palletizing, depalletizing, auto coding, blister packaging, capping, over capping, then leading, closing. So this is the new trends. And as I already told you, the food processing and packaging department, the concentrating on food packaging also. So cleaning, sanitizing, cooling, drying machines are there. All these machineries we studied in, we taught the student at UG level also, and now also in the PG. So when we think on the recent trends in packaging, so this is the recent trend in packaging. Temperature recorders are used to monitor the products shifted in the cold chain. Also, there is a change in the temperature which is recorded into the packet, that packaging unit, or in that pocket. Also, the digital packaging is again digital temperature data loggers are there. So this is the recent trend in the packaging. Time temperature indication are there, which indicate the time and temperature which is experienced by the indicator which is present in the that food. Adjacent foods are there. Then biodegradable plastics are there. So avoiding the wastage, especially solid waste, And as we know that the processing day by day, the demand for the processed food is increased, so the wrappers which are comes into the wastage, and so now everyone is thinking on the biodegradable plastics. So biodegradable films are there, coatings, then synthesized from the organic material by the use of microbial polymers. Some packaging material are edible, for example, the pharmaceuticals, which are we commonly use in the capsule, in that which are made from the gelatin, starch, potato. And now bioplastics are again there. So this is the recent trend in the trades in the packaging. So this is the recent trends in the food processing and packaging. I think that I cover all the points which are comes under the recent trends. So this is the different types of packaging material used in food industry. And just I told you, we from this year we offers the MSc in food processing and packaging. Thank you.
Thank you, sir. If anyone is having any queries, the session is open. Okay, so now I request Ms. Shitoye Pooja to propose the vote of thanks for the session. Over to you, ma'am.
Thank you, ma'am. Good afternoon, all of you. First of all, I would like to thank the director of our institute, Dr. B.T. Jadhav sir, Vice Principal, Dr. V.Y. Deshpande sir, Dr. S.S. Wadikar, Coordinator of Food Processing and Packaging, to give me the opportunity to present vote of thanks on today's FDP session. On behalf of Department of Food Processing and Packaging, I would like to thank our resource person, Mr. P. R. Dure, for their valuable guidance on the recent trends in food processing and packaging. I also making he also making aware about the impact of COVID-19 on food processing and food processing sector. And also, I would like to thank all the participants for your presence. So thank you so much, all of you. Over to you, Jadhav sir.
Session from the Department of Botany, Mr. Y.T. Shinde sir. I request to the department, please run the session third.
Yeah, hello. Am I audible?
Yes, sir. Yes, sir.
Yeah, before we start the session, I would like to introduce the speaker of this session. So, good afternoon, one and all. I am here to introduce the speaker of this session, Mr. Y.T. Shinde, who is working as an associate professor in the Department of Botany, YSI Satara. He has total 36 years of teaching experience at both UG and PG level, and he has worked in various colleges of their Shikshan Sanstha like Pandhapu College, Karad, Ramanand Nagar, Koregaon, Kopargaon, and YCI Satara. He has over 21 research publications at national and international level, and he has also completed one minor research project funded by UGC, and he also written 5 books. So, with this short introduction, I would like to request Mr. Y.T. Shinde sir to begin his talk.
Hello, good afternoon, all of you. I, Professor Y.T. Shinde, Associate Professor, Department of Botany, Yeshwanthaw Chavan Institute of Science, Satara. This institute is autonomous and affiliated also by Shivaji University, Kolhapur. The syllabus of botany is based on the choice criteria today. Lecture is going on. Subkingdom cryptogames and subkingdom phenogames. Subkingdom cryptogames is divided into halophytes, bryophytes, and pteridophytes. Halophytes is divided into algae, fungi, and bacteria. Bryophytes. In algae, there are 9 divisions. There are 5 subdivisions, and in lichen, there are 3 types of lichens, and there are various types of bacteria. Subkingdom cryptogames, high cryptogames in Bryophyta, class Pecopsida, example Trichia, class Anthodopsida, example Anthocerus, and class Musae, example Funaria. In pteridophytes, bryophytes and thalophytes are non-vascular plants, and pteridophytes are vascular plants. In pteridophytes, there are 5 classes. Class Psilophytopsida, example Rhynia horneophyton. Class Psilopsida, example Pteridum. Class Spanopsida, example Equisetum. Class Lycopsida, example Silagenera, Lycopodium. And class Chiropsida, example ferns and Nephrolipis. Subkingdom phenogames is divided into 2 having a seed pendant, and cryptogames is non-having and seedless plants. The subkingdom are divisions. Phenogames is divided into 2 subdivisions. Subdivision gymnosperm and subdivision angiosperm. Subdivision gymnosperm is placed by Bentham and Hooker in between class A dicot and class monocot. Gymnosperm is divided into directly classes: class Psychopsida, class Conopropsida, and class Nitopsida. Class Psychopsida, example Psychus; class Conopropsida, example Pinus; and class Nitopsida, example Nitum, Ephedra, Velvetsia. Then angiosperm in the plant kingdom, angiosperm is divided into 2 classes: class Dicotyledon, class Monocotyledon. Class Dicot is divided into series. Class Dicot is divided into classes, subclasses, series, order, family, genus, and species. The general characters of class Dicot is stem branched, taproot system, leaves with reticulate venation, flower seeds with 1 cotyledons, leaves venation, seeds with 1 cotyledons, and vascular bundles are arranged or scattered in types. These are some characters of class Dicot and class Monocot. Then division Chalophyta is divided into algae, fungi, bacteria, lichen. Halophyta is also called a lower crypto games, and Bryophyta and Pterophyta is also called a higher crypto games. Bryophyta is divided into three classes. Pterophyta is divided into also 5 classes. Into angiosperm, gymnosperm. These are some general characters of general plant kingdom. In general plant kingdom, the plant kingdom comprises on our planet or on our Earth about 3,45,000 varied plant species on our record. In the plant kingdom, Climazomonas plant is capable of moving from one place to another place. This is the locomotion of this plant. But all the plants, the majority of plants have stationary in plant kingdom. In the plant kingdom, there are at about 3,45,000 species, and out of these species, 55,000 species are present in only Brazil, and 35 plant species, that is total number of 35 plant species in Colombia, and the total number of plant species in Mexico is the total number of plant species Brazil ranked first in position and Colombia ranked second in position in the world. There are at about 25 mega diversity nations in the plant in the world. In the plant, lower groups of that is cryptogames, and the cryptogames, the cryptogamic plants are generally called plants. The total number of plant species in algae in the world are at about 27,000 species, and out of these 27,000 species, 2,500 species are present only in India. That is, fungi, the total number of fungi plant species in the world are at about 75,000 species, and in India, there are at about 23,000 plant species of fungi are present in India. The total number of lichens in world are 2,450, and in India are only 22,200 species. 200 species are found in India. The bryophytes in India are at about present in Deccan Plateau. There is large amount of bryophytes are present in Deccan Plateau and in Himalaya. About 6 species are present in the world. The total are 720, and in India are only 64 gymnosperm species are present in India. The total estimated plant species of angiosperm groups in the world are at about 3 lakh plant species, and in India are only 1,500 plant species are present in India. That is general outline of plant kingdom. The classification of plant kingdom is divided into 2: cryptogames and phanerogames. Crypto means concealed and gamus means marriage. In cryptogames, reproduction takes place by spores. The cryptogame is divided into 3 classes: angiosperm and gymnosperms. Lower cryptogames and cryptogames is important because in this plant or in these groups, algae, fungi, bryopteridum, these large groups are present in the cryptogames. In algae, generally The study of algae is called phycology. The green pigment of algae is called green algae. The brown pigment of phycoxanthin of the algae is called brown algae. Then the red pigment, phycoerythrin agent in algae. Then fungus. In the plant kingdom, next point is the fungus. Fungus are non-green chalophytes. Then fungi causing diseases of plants and animals. Then fungi is called parasites. When fungi are present on dead organic matter and causing its decomposition, then it is called saprophytes. Mycorrhizal lichen fungi are called symbiotic. Conidial fungi are called Micromycotina or fungi imperfecta. Plasmodial fungi are called myxomycetes. Flagellated fungi are producing spores or zygospores are called phycomycetes, and blue-green algae, that is BGA, is commonly called cyanophycean algae. The algae, according to gold and wine, is divided into 9 divisions. Out of these 9 divisions, cyanochlorantha is the blue-green algae. Chlorophycophyta is the green algae. Charophyta is the red algae. Then Iglinophyta is Igliner algae. Pheophyta, Pyrophyta, Phycophyta or Phyco. This word is used before phyta. This is the classification of gold and wine. Then in Bryophyta, Heterophyta is commonly called liverworts, because the shape of thallus is liver-like, and the use of this plant is used as liver tonic or liver diseases. Then class Anthoceratops is commonly called hungers, and class Musa is commonly called mosses. Then example of liverworts, Ixia, example Anthoceratops, and Musa example is called Funaria. Then what is the Greek meaning of and phyton means plant? The plant body of the higher cryptogams is divided into root, stem, and rhizome, and the leaves is called pteridophyta. Cyathea and Dixonia ferns are tree-like. Pteridophyta groups are called vascular cryptogams or first vascular plants, because xylem and phloem, these conducting tissue or these tissues are present in the Pteridophyta. But in Bryophyta, algae, fungi, lichen, these xylem and phloem are absent. Pteridophyta are included into 5 groups. Then Rhynia and Herniophyta, this is the fossil pteridophyte. Lycopodium, Selaginella, Equisetum, Nephrolepis are called ferns. Then phanerogams, the groups of flowering and seed-bearing plants are called phanero. Phanero means flowering. Gamus means sex organs. Divided into class dipot is divided into subclass polypetally, gamopetally, and apetally. Polypetally is divided into series, thalamiflory, disciflory, caliciflory. Series and direct divided into order, family, genus. Then subclass gamopetally is divided is also 3 series: bicarpellate, series cypromere, and series imperi. Then order, family, genus, species. Then class apetally or monoclamidy. Subclass but not divided into class. These are general outline of plant kingdom. Then systems of classification. In the taxonomy, Linus, Bentham Hooker, A.P.D. Candolle, Engler and Planter, Hutchinson, these taxonomists classified systems of Linus classified artificial system of classification in 735, and this system is based on only reproductive characters. Hence, this system is called sexual system of classification. Then second system is natural system of classification. Example, Bentham and Hooker systems of classification. This system is based on morphological characters, that is vegetative characters, reproductive characters, and floral characters. Then third system, phylogenetic system. Engler and Planter and Hutchinson classified a phylogenetic system of classification, and this system is based on the phylogenetic or evolutionary significance of the system. Then fourth system is the APG system, that is angiosperm phylogeny groups of the scientist system or taxonomy system. This group is informal international group and contributor. There are at about 19 contributors, and they published 30 research papers in the advanced or APG system is the advanced systems of classification in the plant kingdom. There are 4 versions in the APG system, that is angiosperm phylogeny group system. That is version 90, 198, 206, 209, then 216. These are 2016. These are versions of angiosperm phylogeny group system. Then what is the objectives of these systems? There are commonly 4 to 5 objectives of this course of botany. That is, general objective of this course is to give the knowledge of science, to develop scientific attitude among the students, to develop skill of practical work, experimental work in the laboratory, and concepts of these terms. To make awareness of students to natural resources, there is coals, fossil coal, that is taxonomic knowledge of plants, algae, bryophyte, pteridophyte, angiosperm. That is, in plant kingdom, there is a small group of lichen, and lichen is divided into 3 lichens. Lichens is used for medicinal importance and also spices. Then to improve agriculture and related fields to students. Students should know that agriculture and related fields. Then there are some outcomes of this course. Knowledge of plant science or botany. Students use the knowledge of science of plants and botany to develop scientific attitude, to develop skill in practical work, to understand scientific terms, awareness of natural resources, to plants and related subjects, to related fields, to increase students' knowledge of polytop seeds, to develop work of seed technology, biotechnology, to develop a successful career in seed technology, botany, biotechnology, to inoculate job responsibility in the botany, seed technology, and in agriculture and in technology, to inoculate job responsibility and promote self-employment in botany, seed technology. These are some objectives in botany. Then what is the outcomes of this course of botany to explain features and uses of lower cryptogams. What is the features of lower cryptogams? That is what features of algae, features of that is nostoc and oscillatoria. Of soil or to increase fertility of soil or to yield of because they contains nitrogen. To give the knowledge of plant diversity of lower cryptogams, Anabena, Rixia, Oscillatoria, various types of diseases. Then to explain the basic terms of ecology and environment, biological study of imbalance of ecosystem, interactions of biotic and abiotic factors, like temperature, humidity, light, interactions are indicating the need of biological monitoring, causes of biological imbalance of ecosystem, use of plants in remediation, that is medicinal importance, or any use of plants, handling meteorological instruments, that is handling of maximum thermometer, minimum. Then two regions of India are studies, that is western regions, eastern regions, central Bharat. These are geographical regions or then various types of fossils, impression, compression, petrification, mummified fossils, pseudofossils, that is impression, Tyrophylla, Aerophylla, these are impressions. Then society should know the English of plants, in the concept regarding vascular plants and vascular plants and fossils. Knowledge of higher cryptogams and angiosperms and gymnosperms. Then what is a gymnosperm? Which gymnosperms are used as cultivation in the garden, or which gymnosperms are used to increase beautification of garden, or which gymnosperms are used Pinus, Thuja, these are gymnosperms. Then knowledge of higher cryptogams, that is bryophyte, pteridophyte. The pteridophytic plants are used as ornamentation in the garden. Then angiosperms, various types of angiosperms are used or cultivated, beautification to cultivate beautification in the garden. The knowledge of fundamentals of plant science, knowledge of angiosperm taxonomy, classification of flowering and non-flowering plants, economic importance of medicinal plants, that is ethnobotany. Then what is the outcomes of seed technology? Concept of seed technology, seed identification, crop family identification, concept of plant structure of seeds, morphology of seeds, and in the seed, there are techniques used in crop improvement, that is plant breeding techniques, hand seed manipulation, basic concept of plant exploration, different methods of hybridization, self-hybridization, cross-pollination, self-pollination, taxonomic account of major crop plants, morphology of dicot and monocot plants, identification of crop varieties based on the morphological characters. Students should know tools and equipment in the plant breeding experiment. or in the gardening implement or in the garden, students should know different sort of techniques in plant breeding or techniques of mitosis and meiosis. Then in physiology section of seed, students should know, understand physiological process of seed development, biochemistry of seed dormancy, seed dispersal, seed germination, seed viability, seed germination percentage process, pre-treatment of seed germination, to understand techniques of tissue culture, that is micropropagation, to understand process of hybrid production. This is the importance in the agriculture, to increase production of seed, to perform biochemical test in the seeds, phenol test, seed viability test of seeds in the laboratory, to determine pollen fertility in the plants, to prepare culture media, that is potato agar culture media, Japanese culture media, to study foreign variety of crop plants which is used in hybridization, and to visiting seed production unit in the various fields. Students should know microbes and algae and fungi, and paper second carry plant ecology, and these two papers based on the practical, that is practical first. In semester second, that is paper first carry 6 credit points, second carry 6 credit points, semester second carry 2 paper, paper number 4, biodiversity, paper number 4, plant taxonomy, and practically based on the practical first and practical second, practical first and practical second based on the theory paper third and fourth. In the unit first of paper first, there is introduction of plant and bacteria, unit second algae, unit second fungi. In semester first, paper second, there is ecological factors and adaptations, plant communities and succession, ecological pyramids and physiography, phytoremediation, and semester second, theory paper semester second, theory in gymnosperms, paleobotany. In paleobotany, there is important chapter in the paleobotany geological time scale. Geological time scale is divided into 5 era: Cenozoic, Mesozoic, Paleozoic, Archeozoic, and Proteozoic. These are 5 era, and this era is divided into periods, and periods are divided into systems. In paleobotany, there is an important chapter of fossil formation process, that is impression, compression, petrifaction, cast, pseudofossils, and pole vaults. These are some important points included in our syllabus of practicals. Then in seed technology, there are 4 papers. Paper number 1, seed development and morphology. Paper number 2, seed physiology and seed production. These 2 papers carry, semester number 1, these 2 papers carry 4 lectures and 4 credit points. Practical carry 2 credit points. Same as botany. Seed technology, semester second carry 5 lectures and 4 credit points, and 2 practical credit points. Then paper first, introduction to seed technology. First unit, introduction, crop families and embryology. These families are Fabaceae, example Phaseolus, family Brassicaceae, example Brassica, family Solanaceae, example brinjal, family Poaceae, example maize. Then structure of anther and development of male gametophyte, development of female gametophyte, endosperm formation, reproductive biology, apogamy, allogamy, pollinating agent, apomixis, polyembryology, effect of environmental factors on soil biology. These are 2 papers included in our syllabus of semester first and semester second also included two papers. Paper number third, physiology of seed development, seed dormancy, seed germination, concept of advanced seed. Then paper fourth, seed quality concept, hybrid seed production, genetic purity of varieties, procedures. These are 4 units in semester second, paper 4 in the seed technology section. In MSc, there are is also credit-based system syllabus, duration of the degree 2 years, pattern is CBCS or choice-based credit system, 4 lectures per week, but this paper or this code carry 4 credit points. Total semester first of MSc carry 24 credit points, same as semester second. Semester second carry 5 papers: MBT 201 cell and molecular biology, MBT 202 taxonomy of angiosperms, MBT 203 plant pathology, MBT 204 MBT 205 plant physiology, MBT 206 practical paper, and MBT 207 practical 4 based on MBT 205 and 204, and extra 3 credit points, that is 2 credit points, tutorial-based day-to-day performance. And 1 credit point MSc for tools and techniques.
माळी सर, सर लेफ्ट झाले का? हॅलो माळी सर.
introduction of botany.
You again say.
आता काही काही झालं झालं की
My
सर झालाय जॉईन आता येतोय आवाज.
आता नाही झालं. Thank you for attending the lecture झालं की and listening the lecture. Botany topic or general botany, introduction of plant kingdom. Thank you.
There was a small technical error. The internet connection went off, so sir could not conclude in the effective manner, but he was already at the end of his lecture. As requested by Jadhav sir, I would end this session by the vote of thanks. I am very thankful to Mr. Vaiti Shinde sir for taking our precious time and telling us about the latest updates in botany and paleobotany in particular. And I thank all the joining in, and I hope the lecture was good enough and
Thank you, ma'am. Deliver the different topics, deliver the lectures on the different topics, and I'm also thankful to all the head of the departments and senior faculty members. I'm also thankful to the Honorable Principal Dr. B.T. Jadhav sir for supporting. So once again, thank you all of you, and with the permission of organizer, conclude the this. Thank you very much.